Also, as I explained, it gets better because the linear DNA molecule is very stable because the backbone is on the outside and is not chemically reactive. The chemical reactive parts are on the inside and are protected one with another. So it's a very stable way of storing that information. However, it can't do anything because it's so stable. It cannot do anything. But remember the central dogma. You go from DNA to RNA, and then you go to protein. And remember what I said about protein, and I'm going to just show you the same slides again. Now, you still have a polymer. It still reflects information. Each of those amino acids, what you put in there and the order is determined by the base sequence in the DNA. But now everything is facing outwards, and all the chemistry is facing the environment. So you have oxygen, you have nitrogen, you have positive charge, negative charge, much less stable, but now it can carry out chemical reactions. So what you do is you are turning stored information in nucleic acid into chemical actions in protein which are needed for life. So you have a storage device which is stable, and you have a protein which are reactive and carry out the chemistry of life. This is really fundamental in understanding life, because you can solve the problem of information storage and you can solve the problem also of chemical action. Second example, relevant for information and making sense of biology and information, is seen with gene regulation. This was shown by Jacob and Monod, who I knew, or at least I knew Jacob quite well. And they did it by doing beautiful genetics with bacteria, looking at sugar metabolism in bacteria. And what they did by abstract genetics is discovered negative feedback control. What is negative feedback control? Well, this is a governor from a steam engine. Now, you may be like me. I sort of understand most machines built before 1900 and almost nothing built after 1900. I took a picture of this in New Zealand, actually. It was in a steamship, and I went down and photographed it because it was such a beautiful thing. It was found originally on James Watt's steam engines, and we understand it. The spindle rotates. The balls are thrown out. They lift a valve, and that closes steam off from going into the engine, so it slows down. Then the balls go back in again, and they let steam back into it, and it speeds up. And this is essentially homeostatics. It's maintaining a certain, in this particular case, rotation of the particular spindle. And what Jacob and Monod did was to use this principle to describe regulation. And here we're looking at something, in fact, not to do with gene regulation, but it's the same principle as what they described, where we have a chemical A turned into chemical B, turned into chemical C. There's enzymes, which are the green arrow, and as C accumulates, it switches off the enzyme that is catalyzing A into B, and therefore you inhibit making B, you inhibit making C, and then C drops in level, you lose the inhibition, up comes the enzyme between A and B, and you go back again. And what this is doing is maintaining, by negative feedback, a constant source of material. If we look at the bottom one, we have positive feedback loop, which is the complete opposite. When you accumulate C, it switches on the enzyme going from A to B. So once you've started this, it turns into a switch, and you cannot go back. You get the principle. And life's metabolism and gene regulation is built on these simple principles of this type of feedback control. Now, you can put them together in all sorts of complicated ways, and a metaphor is an electronic circuit that you see here. And these circuits, which we find in life, can produce negative and positive feedback, switches, timers, toggles, oscillators, all of which are playing a role in life. Now, it's rather special. Just like the chemistry is special, this is special. Because when we think of, for example, computing, which is, of course, the same principle, you have hardware and software, yes? The hardware can't change. The software, you can make the hardware operate in different ways, but the hardware is wired in. In biology, it's cleverer. It was the term was used by Dennis Bray, he is a systems biologist. He used the word wetware rather than hardware, because the communication between the different regulatory steps is carried out by molecules diffusing through water. And this means you can rewire the hardware by directing the chemicals to go to different places. So not only can you reprogram it through, if you like, changes in software, you can also change it through changes in hardware. We do not yet begin to understand this complexity, but it's important. Now, networks can be very complex. We see at the top here how we like to think about things, or to be more precise, how men like to think about things, which is linear pathways going from A to Z. But the truth is, in biology, it's much more complex. And whereas we all intuitively understand what's happening at the top there, we don't at the bottom. It's just too difficult. That is because of the next idea I shall talk about, that of evolution by natural selection. Simply means that you add things on to something and make it just more complex. Not necessarily the simplest way to do something, but simply a way of actually doing it. But I want to mention one more thing that we don't often think about in these pathways, that if we introduce dynamics into a pathway, that is, changes in time, it becomes much richer. And this is a simple example of it. We have a single, at the bottom there, pathway, and now we're pulsing information down, and either frequently or infrequently. And the output can be different, depending on the oscillation, the frequency. This has already been seen. Why am I stressing this? Well, I'll tell you why. Here we see a metaphor for it. Can you see the traffic light? If it's a simple on-off signal, it's either green or red. Not a lot of information. If you introduce dynamics, you can produce the Morse code, because you're pulsing information. And now you can write the works of Shakespeare. Now, I'm not suggesting the cell is writing the works of Shakespeare, but I am suggesting that we haven't got to beginning to the bottom of how this is all operating in cells. They are absolutely extraordinary and are the basis of how living things work. So what I've explained to you is the way it's working is through chemistry plus physics and then management of information. And that's key to understanding what life is. Now, my final idea. I can't stop it. This is the most beautiful idea in biology. Evolution by natural selection. It's got two aspects. Life evolves, and a major mechanism of it is natural selection. It's a beautiful idea, which we mostly associate with Charles Darwin, who you see on the right. But actually, the idea of evolution, that is, of living organisms changing over time, was not Charles's idea. It had been talked about for a century before by French scientists like Lamarck, for example, but also Erasmus Darwin, who's shown there on the left, who was his grandfather of Charles. Now, Erasmus is a rather entertaining character. So he was a poet. He was a doctor. George III asked him to be his physician more than once, but he was a Republican and wouldn't do it. He was in favor of female education, and he set up a girls' school and wrote a book on how women should be taught. It was the first female school in the UK, so I'm told. And he got into trouble with his local dean. And he had on his coach the motto, "Everything from shells." What did that mean? If you open up a shell, you see a sort of formless blob in it. Yeah? And what he was arguing is all life came from formless blobs. And because he actually only charged rich patients, he didn't charge poor patients, he had to paint it out. And he wrote a number of verses about life-changing evolution. But he had no mechanism. And that was led to his grandson, Charles, over there, who gathered enormous evidence, first of all, the fact that, based on fossil evidence that there had been evolution, and secondly, during his voyage on the Beagle, and secondly, that he proposed a mechanism. And the mechanism was natural selection. And it goes a bit like this. Actually, completely like this. I don't mean a bit like this. Within a species, there are variants. They have differences. These are due to inheritable differences. They're not ones where if you grow a plant in the sun or not in the sun. They are inherited differences. Those variants, genetically inherited ones, which are most successful, reproduce more, therefore pass on more genes to the next population in the next generation. And therefore, there is selection for changes in characters. And over time, that will lead to speciation and therefore evolution. And it also accounts for why you can get exquisite adaptations. And that we see here. He studied these finches in the Galapagos and came to the conclusion that they were beautifully adapted to whether they fed on insects or broke nuts. And I put up there a series of tools which are also perfectly adapted for what they do. So, two things. He argued that there was evolution, so all life was related. This is the one figure in The Origin of Species, related by descent. And secondly, there is beautiful adaptation. And the reason why this is controversial is because it can lead to apparently purposeful behaviors. And if you have purposeful behavior, normally it's easier to describe that because somebody's made it. And there's the famous story of the Reverend William Paley finding a watch on the path. And when he saw the watch, he said this had to be created. And when you see animals and plants perfectly adapted, he argued, therefore they had to be created. But what Darwin's idea of natural selection showed is that you could evolve by natural selection, apparently purpose, without having to postulate a creator. It would come naturally from that. Now, how can it actually occur? Well, it occurs because life reproduces. It has a hereditary system based on genes, which determine what life is like, and it has variability upon which natural selection can work. And there I've turned this into single cells. And what you see here is a cell, I hope it's visible behind me, which starts being orange and ends up being red, at least on the right-hand side. Do you see that? Okay. The idea is this, that you have genes, they're replicated, you pick up an alteration, what we'd call a mutation, and that leads to the coat of the cell being red rather than orange, and red cells have a reproductive advantage, so over time, you replace the orange population with a red one, okay? So that's a simple evolution by natural selection. And what we have here is everything you need to evolve. You have, and that's seen in cells. You have genes of the hereditary system. They show variation because they're not copied completely precisely or they're damaged by external X-rays or sunlight. And then that can lead to evolution by natural selection. I want to finish this part with a quote. This is the old Charles Darwin, the one that we tend to think of. And it's the last sentence of The Origin of Species, because he wanted to show that he could produce laws in biology just like Newton could do it in physics. And he states, whilst this planet has been gone cycling on according to the fixed law of gravity, obviously reflecting Newton, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. It's a beautiful ending to a book.